Well, hello, and welcome to Bio 212, Anatomy and Physiology 2 at Norwalk Community College. And what we're doing is we are beginning Chapter 29, where we're looking at development and inheritance. The chapter is essentially two pieces, the first of which is what happens at fertilization through birth or parturition, and then can you examine the consequence of fusing two haploid cells together? What are some of the introductory levels of understanding of gene expression inside of now the new fetus, such that it can survive in the environment that it's been born into? So having said that, do a quick little sketch here. Remember, spermatocyte, A. Okay, let's put a tap hat on you, yay. Top hat plus secondary oocyte that was ovulated from the ovary, right? So this is 23 chromosomes here, not 23 pair, 23 chromosomes here. Together, they're going to give us a zygote. That's what the big Z stands for, and that's 23 pair of chromosomes now, all right? And remember, they're all stored inside a single nucleus. So if I put this over here, we're going to put the nucleus inside there. And remember, the DNA, of course, of your cells are the instructions to make the proteins that are required for a cell to live. So if this is one cell and you are who you are today, it means you've undergone trillions and trillions of cell divisions to actually get to where you are right now. What this chapter does is it begins with this concept of, well, what exactly is fertilization? Let me change colors here for just a second. Go something like that. And I'm going to put an acrosomal cap around the outside of this thing, and I'm going to change and put, I don't know, let's put blue just for the fun of it. We're going to put a blue layer around here so that we can begin to break down some of what's going on here. Okay? So what you're looking at, of course, is the genetic material from the spermatocyte, and the secondary oocyte will merge into that zygote there. And usually occurs inside that fallopian tube, just like I told you inside class. What's happening, though, while the spermatocytes are inside the vagina, right? So ejaculation occurs. Remember, parasympathetic nervous system has to be activated first, then sympathetic nervous system for ejaculation to occur, and that will yield the capacitation, meaning once inside the vagina, it now becomes functionally different, so it can now fuse with the oocyte. Because that's what's going to actually have to happen here is that acrosomal cap will begin the process of digesting that corona radiata and the zona pellucida. And remember, inside lecture, inside class, I said there's a receptor here, and the receptor is called the ZP3, meaning zona pellucida 3. And what's going to happen, of course, is that that's what actually gets the spermatocytes to get to that secondary oocyte and then break down that glycoprotein layer. Okay? It's the acrosomal enzymes and strong movements, meaning the flagellar tail, pushing the actual spermatocyte now into the secondary oocyte. And if you remember properly, by the time you finish chapter 28, you have a secondary oocyte that is arrested before the end of meiosis II. So you can imagine that this will have a series of chromosomes aligned along the metaphase or anaphase structure. Okay. Having said that, let's actually get rid of the drawings here. Okay, let's go over here. And you're going to see pretty much what I was trying to draw. Notice here inside your textbook, they have it arrested at the anaphase 2 portion of meiosis II. But one spermatocyte has gotten through. This layer right here is the zona pellucida, which is a glycoprotein structure, and it's surrounded by those granulosal cells. And those granulosal cells, of course, are what is creating or was supporting this as it was growing inside the follicle. Okay? A couple of cartoon, photomicrographs in there for you to see that. What's going to happen, of course, is a series of mechanisms have to stop more than one spermatocyte from getting in. And that, of course, would be called polyspermy. So you actually have, let's go and do that. Control C, go that, let's go that. Okay. Where polyspermia would be more than one sperm actually getting in. So a fast block, okay, is actually going to create the mechanism that stops other sperm from actually fusing, okay? The slow block, which is going to have, how do I say it? So this is going to be a calcium mechanism. Let's put just calcium, Ca2 plus here, mechanism, which actually creates a wave that covers the surface of this. And what's going to happen now is the slow block to polyspermy actually causes the hardening of the entire zona pellucida. Okay? So just put an underline underneath that. And now it's going to finish. Okay? The meiosis. Now that it's been fertilized, you're going to pair up the chromosomes, and you're now going to finish that, and you're going to use the polar body that's going to be broken down and then used as an energy source to actually get through to the next stage. So what's going to happen, of course, is the male pronucleus and the female pronucleus divide, so you end up with 46 chromosomes in total. Now, you're going to have, once you actually have this really large secondary oocyte that has been fertilized, so therefore it's a zygote, it's going to actually begin division, okay? And what's going to happen is each successive division takes less and less time. But remember, I'm going to draw this for you. We started with essentially a secondary oocyte that had been fertilized. Now we actually have a nucleus, and we still have our zona pellucida around this thing. So let's pick that blue, put that blue inside here. And the part that we want to make sure you understand is this is one big cell, that zygote. And every time it's actually undergoing a division, then it's going to become two cells. You probably shouldn't have used that light blue there, right? But you get the idea. Each time it's staying inside the small structure of the zona pellucida, and it's making a series of smaller and smaller cells. And that's why it's called a blastomere. The consequence of this is you actually make that morula that you saw, or morula, that you saw inside lab. Okay, it's a solid ball of cells. Still actually has a zona pellucida around it, and it's the same size, roughly, as the original zygote that was fertilized. Okay. Why do we care? Well, we care because we actually have to get this thing to now move. Because everything's been trapped, what Mother Nature did is she created a system where you trap fluid inside of that morula, and now what's going to happen is you actually have the increase in pressure inside of that. So that's going to actually create a cyst. So it's the actual formation of metabolic water that actually drives the creation of this cyst. So let's go like that, underline, fantastic. And it's going to take about 4 to 5 days to use the cilia of the fallopian tubes to actually move to where it's supposed to move to. And inside class, I told you that the PEG cells, okay, as well as uterine milk, are going to make glycogen-rich secretions. They're going to keep this whole thing alive. Now, what's going to happen by the time they skip over some steps here, right? Which is both frustrating, but I mean, we don't want to spend our entire day talking about this stuff, right? What I want you to see is by the time it actually gets to a blastocyst, it's now fluid-filled, okay? So it has a blastocele, and there are 2 layers inside of it. So our inner cell mass will have, okay, that inner cell mass is first called the embryoblast, and you'll see that in a second. And the trophoblast, okay, will be the outer portion, okay? And what's going to happen is about 5 days after fertilization has occurred, that zona pellucida will actually break down. Okay, it says it hatches from. Essentially, remember, it hardened. So now now you actually have to move out of it for implantation to occur. Okay, so to see this, I was going to draw it, but that's going to take too much time. So to see this, here is the blastomeres that will end up cleaving, and notice this solid structure out here is still the zona pellucida. Notice you actually have smaller and smaller cells, but if we cut open this blastocyst, it's now fluid filled, and you have this embryoblast on the inside. It's fluid filled, so therefore it's a blastocyst cavity, but the entire outer portion now makes up the trophoblast. And the significance here now is that we have a mechanism to orient the developing blastocyst to place inside the uterus, and this is how it does it. It's going to find its way to the fasciculatus, right? So, but you're going to note that the functionalis. What am I saying? Sorry. Functionalis. What's going to happen, of course, is it moves along. It uses the orientation of the mass change to position itself, so the inner cell mass will divide its way in. And what's interesting that I want you to pay attention to is because we're digested its way into the stratum functionalis, we change its name. Now it becomes the decidua, okay? And because it's covered over, it's called the decidua capsularis, where it digested its way in. It's the decidua basalis, okay? And what's going to happen, because fertilization has occurred, the endometrium is going to become more vascularized, okay? Which makes sense. We have to get oxygen there, and we have to remove the metabolic waste products. So this is almost exactly what you want to have happen. You're creating an environment that's going to allow now for all of these cells to continue to divide. And from lab, you might remember that this is going to be the hypoblast, the outer portion here, and the epiblast will be on the inside part here. And that amniotic cavity will now become much more prominent. Before we get there, though, let's take a look at what's actually going on. It's rolled. It's now positioning itself, okay, and it's going to digest its way in. So if fertilization occurs somewhere around here, cleavage occurs, you're going to actually put that blastocyst now inside the uterine lining, where this is the basalis here, functionalis here, and as you digest the way in, remember, we now call it a decidua. So we're going to actually have the development now of that trophoblast, and the significance here, of course, is that it's going to become buried inside the endometrium, and because it's become buried, the structure now begins to make a hormone, which means from this point forward, you now think of the uterus that has a fertilized blastocyst inside of it as an endocrine gland, because it's going to make human gonadotropin, okay, and it's going to maintain the corpus luteum, put that bold underline, okay, inside the ovary so that it can continue to secrete two hormones, estrogen and progesterone, okay? And in doing so, it makes sure that the vascularization of the uterine lining is maintained, okay? So far, before we get too much into some details here, what we've spoken about is fertilization has occurred. You understand the mechanism by which you've regulated that, okay, meaning fast and slow polyspermy block, okay? And then, because you now have a zygote that is trapped inside the zona pellucida, all those cells become smaller and smaller, and they're moving towards the uterine lining, where they will digest their way into the basalis layer here. And once inside that basalis layer, it begins to secrete human chorionic gonadotropin, which will then, using the circulatory system, target the corpus luteum, which is inside the ovary, right, to continue to make estrogens and progesterone. Imagine, if you will, you had to draw that, or you had to discuss it. I mean, think about what's actually going on here. You actually have to use the circulatory system for all of this, okay? So, and a moment ago I pointed out to you, you actually have a bilaminar embryonic disc, okay, that hypoblast and that epiblast. And you'll notice here, inside lab, when I was talking with each of you, I said, remember, we have three layers we actually have to get to: endoderm, mesoderm, and ectoderm. But that hypoblast becomes the endoderm. The epiblast becomes the ectoderm. Okay? And what you're going to see, of course, is that you're going to now begin building around these tissues so that you actually have an amniotic cavity that is below it. Okay? An amniotic cavity that is below it. Okay? Let's see if we can see that for you here. Okay, so here's the digestion. We begin to digest our way in, and remember I said the trophoblast becomes two populations of cells. Well, there they are. There's the cytotrophoblastic cells that make up still the remaining circular structure, and the syncytiotrophoblastic cells. These guys are called syncytial cells because they fuse, and they're literally fusing and digesting their way through, and they're creating sinusoids, we call lacuna inside lab. And those lacuna are adjacent to blood vessels, so diffusion of materials from the maternal circulatory system to these lacuna will support the developing growth of cells here. this is this is what, 9 days, and this is 12 days, and you can actually see what's happened is you still have that bilaminar disc, but you can actually see here in red, this extra embryonic mesoderm is now beginning to move around this. So you actually have ectoderm, endoderm, and mesoderm. You'll see why that's important in a minute. Okay, so because we need to actually make something to keep all of this alive, we will make the yolk sac will be made from the hypoblast, okay? And what's going to happen, of course, is you need to have supply of nutrients to the cells, source of blood cells. Whoa, where'd that go? Okay, where are you? Here you are. Nope, I'm still ahead of myself. What happened there? There we go. Okay. You have primordial germ cells that are going to be inside that yolk sac. We've spoken about those, okay, that are going to migrate to the gonads and form gametes. Part of your digestive system, the gut, is there. It also functions as a shock absorber to prevent damage as the body is moving around. And there's also desiccation. Okay. Those sinusoids were made from those syncytial trophoblasts. Okay, and you're going to actually now form a chorion. And that chorion now begins to produce human chorionic gonadotropin. Okay? So we saw that a moment ago, right? Let's see what we got back here. Okay? So the blastocyst now is going to secrete human chorionic gonadotropin. And now that we actually have further development, let's see what I can do here. The chorion is now going to begin to make more human chorionic gonadotropin. So we have a concentration change inside the circulatory system. But this also means that you're going to become less dependent upon the ovary to maintain the placenta. What's going to end up happening is as the chorion becomes the placenta, it will make more and more human chorionic gonadotropin, and therefore you will no longer need the ovary for the production of the estrogen and progesterone. By the time we get to the 6th week, right? Okay. Gastrulation is the major mechanism by definition. And what I mean by this is don't memorize all the steps of gastrulation. Okay. What's going to happen, of course, is you actually have the bilaminar disc transforms into a trilaminar disc. And this is how you make ectoderm, or what ectoderm becomes, mesoderm, and endoderm. Okay, and inside your text, definitely inside class, we've gone over, or we will go over, all the details. You're going to notice the list inside AMP2 is definitely more precise than the list inside AMP1 for what arises from ectoderm, mesoderm, and endoderm. As we actually pass through this, here's what's happening. Here's that bilaminar disc. You're going to actually begin to separate this. And if you take this and cut this sideways, you're going to see now where mesoderm begins to come and fill in between these layers. So epiblast, right, is now going to be called ectoderm. The hypoblast is the endoderm. Okay. And now you're going to actually see the tissue move in between those spaces. That actually finishes up gastrulation, right? So when we're going to actually, by the time we actually finish gastrulation, we now have a structure that's big enough so that you actually have the other way that I describe you guys. You guys are essentially a tube inside of a tube, right? And if you look at the human body, you see that there's a series of tubes throughout the body. We call them ductal structures and all that sort of stuff. But take a look at what we actually mean. You have now formed the anus. You actually have the reproductive tracts as well as the urinary tracts. And what this does is this allows the developing population of cells to have a mechanism by which you can transport materials inside the developing body. Okay? Wham. Here's that trilaminar disc there. And this is the tube inside of a tube, which is eventually going to be the separation between you, okay, and the outside world. We have to put in a nervous system inside of all of this, and that's what neurulation is. And you'll notice if this is a developmental biology class, I'll be going into a lot more detail. But all I really want to do is get us to now the fact that we actually have a developing structure. The first thing that actually we spend any time thinking about are how do you actually create neural folds, develop a brain and spinal cord, and then give rise to all of the cranial nerves, the ganglia, the autonomic nervous system, as well as everything else that's going to be innervated by these. And that's pretty much where we're going to stop, because take a look at what's actually here. You have the prosencephalon, the mesencephalon, and the rhombencephalon. And I want to see if we actually have a picture of it down here. We do not. Okay, so essentially inside class, you want to know the definition of what is gastrulation and why is it different than neurulation. That's a pretty good place to stop. Okay, so we'll pick up. We'll start here in the next video, and then we'll move into the second phase, which is, well, what happens when we actually now need to build a placenta to support the growing number of cells.